Stay home.
Stay home.
Stay home.
នៅផ្ទះ
Stay home.
อยู่บ้านเด้ออยู่โรงพยาบาล
Stay home.
อยู่บ้าน
Stay home.
Stay home.
Stay safe, ASEAN.
I have a dream of what I wanna be. I wanna be free. That's what my heart keeps telling me.
Give me your hand, we'll find a way. There are people out there that feel the same. And if we help each other, I just know we can succeed.
We are one, but we are different. We're from all around the world. We share the same sun, we share the same oceans. We are every boy and girl.
Come together and share our dream. We'd be among the stars, and we know that it's destiny. We are unity. We are unity.
I do believe, oh, I believe in destiny. It's up to me to make it into reality.
Give me a hand, we'll find a way. There are people out there that feel the same. If we help each other, I just know we can succeed.
We are one, but we are different. We're from all around the world. We share the same sun, we share the same oceans. We are every boy and girl.
Come together and share our dreams. We'll be among the stars, and we know that it's destiny, that we are unity.
No matter where you're from or who you are,
If we can find a common place to be, then we are friends.
we are one, but we are different. We're from all around the world. We share the same sun, we share the same oceans. Ooh. We are one, but we are different. We're from all around the world. We share the same sun, we share the same oceans. We are every boy and girl. Come together and share our dreams. We'll be among the stars, and we know that it's destiny, that we are unity. We are unity.
We are unity.
Destiny, that we are unity. We are unity. love Malaysia now and forever. Different places everywhere. So Asia is surely here. This beautiful Malaysia. You love the colors of Malaysia, where the sun loves to shine on sandy beaches and clear waters with smiles of friendly races. This land so beautiful, it steals your heart away. This land is paradise, only a smile away.
Stay home.
Stay home. Stay home.
នៅផ្ទះ
Stay home.
อยู่บ้านเด้อดูเรื่องมา
Stay home
อยู่บ้าน
Stay home.
Stay home
Stay safe, ASEAN.
I have a dream of what I wanna be. I wanna be free. That's what my heart keeps telling me.
Give me your hand, we'll find a way. There are people out there that feel the same. And if we help each other, I just know we can succeed.
We are one, but we are different. We're from all around the world. We share the same sun, we share the same oceans. We are every boy and girl.
Come together and share our dream. We'd be among the stars, and we know that it's destiny. We are unity. We are unity.
I do believe, oh, I believe in destiny. It's up to me to make it into reality.
Give me a hand, we'll find a way. There are people out there that feel the same. If we help each other, I just know we can succeed.
We are one, but we are different. We're from all around the world. We share the same sun, we share the same oceans. We are every boy and girl.
Come together and share our dreams. We'll be among the stars, and we know that it's destiny that we are unity.
No matter where you're from or who you are,
If we can find a common place to be, then we are friends.
we are one, but we are different. We're from all around the world. We share the same sun, we share the same oceans. Ooh. We are one, but we are different. We're from all around the world. We share the same sun, we share the same oceans. We are every boy and girl. Come together and share our dreams. And be among the stars. And we know that it's destiny that we are unity. We are unity.
We are unity
Destiny that we are unity. We are unity.
Assalamualaikum. Hello and welcome to the second AACLS live webinar series on the current COVID-19 pandemic. I'm Rafida Muhammad, your MC for today's event, hosted by the Malaysian Institute of Medical Laboratory Sciences, MIMLS, under the auspices of ASEAN Association of Clinical Laboratory Sciences, AACLS. This event is exclusively brought to you live from Kuala Lumpur, Malaysia by ChemoPharm and MindRay. Thank you for joining us today, and we do hope that everyone will stay online with us right till the end. As most of us are well aware of, the theme for today's webinar is the role of laboratory during COVID-19 pandemic, a shared experience. Before we begin our webinar sessions, let's have a quick rundown to today's program. We will start with the president of MIMLS, Mr. Q Jai Kang, giving the welcoming message, and followed by the opening speech from the president of AACLS, Dr. Miswar Fatah. Then I shall pass over the program to Mr. Woon Santong, the past president of AACLS, who will be our moderator for today's webinar sessions. There are 4 interesting talks coming your way, which will be delivered by our distinguished panel of speakers, beginning with Ms. Havinda Kaur of MIMLS Malaysia, Mr. Tin Tun of SAMLS Singapore, Dr. Zainun Zaini of BAMLS Brunei Darussalam, and lastly, there will be an industrial talk, which will be delivered by Mr. Christian Bozak from Drager, Singapore. In order to make this event run smoothly, we do hope everyone will adhere to the housekeeping message shown earlier. Let me go through it once again. Please ensure that your microphone is muted at all times during this webinar. Once the session begins, you may pose your questions by clicking the Q&A box on your screen. Please do not forget to state your name and country in the Q&A window along with your question. The speakers will then respond to as many questions possible during the Q&A session right after the industrial talk. During this Q&A session too, we will post a link in the chat box for those who want to have their certificate of attendance for today's webinar. Towards the end of the event, we would really appreciate if all of our participants in Zoom could spend a couple of minutes to answer the event poll. Thank you for your attention. It is now my great pleasure to invite the president of MIMLS, the Honorable Mr. Q Jai Kang, to give the welcoming speech. Ladies and gentlemen, please welcome Mr. Q Jai Kang.
Thank you, the Master of Ceremony. Dr. Miswar Fatah, President of ASEAN Association for Clinical Laboratory Sciences, AACLS. Moderator, Mr. Woon Santong from Malaysia. Distinguished speakers, Ms. Havinda Kaur from Malaysia, Mr. Tin Tun from Singapore, Dr. Zainun Zaini from Brunei Darussalam, and Mr. Christian Bozak from Drager, Singapore. Organizing committee members, ladies and gentlemen. My name is Q, and it's my privilege and pleasure on behalf of MIMLS and organizing committee of this event to welcome you. We are delighted to have you with us to participate in the second AACLS live webinar. Thank you for joining. In line with MIMLS' commitment in promoting the developments and advancement in medical lab sciences, we are pleased to host this webinar with theme, The Role of Laboratory During COVID-19 Pandemic, the Shared Experience. 4 speakers from different countries will be sharing their expertise and insight about COVID-19, laboratory experience, and newborn jaundice screening. I certainly hope and believe that this webinar will help the medical laboratory professional to obtain new knowledge and practices in medical laboratory sciences. And before I hand over to the MC, I want to say once more on behalf of the webinar organizing committee, welcome. And it's wonderful to see so many of you here. Stay safe and healthy during this pandemic. Thank you.
Thank you, Mr. Q. To officiate today's webinar, I am pleased to invite the president of AACLS, the Honorable Dr. Miswar Fattah, to give the opening speech. Ladies and gentlemen, please welcome Dr. Miswar Fattah.
Thank you very much, Mr. Rafida Muhammad. I am very excited to meet online with ASEAN clinical laboratory scientists today. Hello and welcome to today's webinar, the second ASEAN Association of Clinical Laboratory Scientists, AACLS. Thank you for taking time out and being here today. This webinar will taking about the role of laboratory during COVID-19 pandemic, and this is based on shared experience. I would like to say thanks for host for this event, Mr. Keo Ik Kang as President MIMS and all committee. I know they're very active and busy to prepare this webinar. And thank you for our senior figure in ASEAN Clinical Laboratory Scientists and one of AACLS past president, Mr. Woon Sung Thong, as a moderator today. And thanks for all speakers, Harvinder Kaur from MIMS Malaysia, Mr. Tintun, all friends from SAMS Singapore, Dr. Zainun Zaini from BAMS Brunei Darussalam, and Mr. Christian Boza. In just a few months, we take us in how the coronavirus changed our life and how much suffering it has caused across ASEAN and the world. The COVID-19 pandemic presents extraordinary challenge that taught us all in our profession and professional life. Part of professional in laboratory medicine, we have a critical role to fight this pandemic. Thanks for all participants who joined today, all clinical laboratory scientists in ASEAN. I hope you always be healthy and fight COVID-19. Enjoy this webinar. Thank you, Mr. Rafida.
Thank you, Dr. Miswar. Ladies and gentlemen, I would like now to proudly introduce to you our moderator, who is personally my mentor in this field of CLS, Mr. Woon Sung Thong. Mr. Woon is a graduate from University of Malaya and began his career as a medical laboratory technologist in the University of Malaya Medical Center, Kuala Lumpur from 1968 to 1999. During his tenure there, he was involved in the training and teaching of MLTs, medical students, biomedical sciences students, as well as doctors pursuing the Master of Pathology. He's also very active professionally. He was founder member of the MIMLS, Secretary of the Laboratory Quality Assurance Scheme, LabCAS, which is joint activity with the College of Pathologists Malaysia, and also a technical assessor for ISO 15189. Regionally, he was instrumental and a founder member of the formation of the ASEAN Association of Clinical Laboratory Sciences, AACLS, that is in the Southeast Asia region, and the Asian Association of Medical Laboratory Scientists, AAMLS, that is for the whole continent of Asia. He was past president of AACLS and a former director of AAMLS. Mr. Woon had also held the post of head scientific affairs Sysmex Asia Pacific in Singapore for several years. In 2017, he was awarded AAMLS Award Professional category during the 5th Congress of AAMLS in Busan, Korea. Currently, he is a trainer for phlebotomy and a frequent speaker for CME. And we are very fortunate to have Mr. Woon with us today. With the enthusiasm of the organizing committee, our panel speakers and our supporters, we do hope that our audiences will get the most out of today's webinar. Therefore, without further ado, I will now pass over to the moderator, Mr. Woon Sung Thong. Over to you, Mr. Woon.
Thank you, Rafida. I'm Woon, the moderator. Good morning, AACLS members and friends. Before I start the session, I would like to wish all our Muslim colleagues and friends Selamat Idul Fitri. Welcome to the second leg of the AACLS live seminar. Just 4 weeks ago, the Tauki of Indonesia hosted the first webinar, and now it's our turn. I'm sure that during this time of the COVID-19 pandemic, we laboratorians have been put to test on what we can contribute to combat this battle with the coronavirus. So let me begin by asking Havinder Kaur, Havinder, to start the session. Havinder Kaur started her career as a medical laboratory technologist in medical microbiology in August 1999. Havinder's interest in biosafety and biosecurity was sparked during the Nipah virus outbreak, culminating in a Master of Science degree in Occupational Health and Safety Management from UUM. Havinder has also undergone training and certification by CRDF Global and is a registered biosafety professional. Besides possessing knowledge of many quality issues in the clinical laboratory, including ensuring compliance with all regulatory requirements, she is also well versed in conducting gap analysis, developing, implementing, and maintaining a quality management system in the laboratory. Her work with researchers on various infectious diseases has resulted in ISI indexed publications and poster presentation at numerous scientific conferences. She also had the honor of being an invited guest speaker at seminars and conferences to share her experience and knowledge. Havinder is also involved in teaching of the medical laboratory technologies students, biomedical as well as the master pathology students. Havinder is on the working committee of the Occupational Health, Environmental and Safety Services of PPUM. That is the University of Malaya. She is also on the working committee of University of Malaya Institute of Biosafety and Biosecurity. Havinder's credentials also include being appointed on the national level as a committee member involved in the Allied Health Profession Act for the medical laboratory technologies profession in Malaysia. Ms. Havinder, the floor is yours.
Thank you very much. Good morning to all fellow speakers as well as dear guests. On this wonderful morning, I'll be talking about the use of other biomarkers for the detection of COVID-19, as well as how is it that the research laboratory came together during this pandemic, our experience in Malaysia. Right. Now, there was a very great rationale for adopting other biomarkers because, as you know, currently the gold standard is the nasopharyngeal aspirate, but that produces a lot of aerosol generating particles that will infect the healthcare. By adopting other biomarkers, our sampling procedure can reduce risk to healthcare, and discomfort to the patients. It can also improve transmission prevention, especially for healthcare workers, as the current procedure is aerosol generating and highly laborious. Now, the current situation, there have been tremendous advances made for the in vitro diagnosis of coronavirus caused by SARS. Currently, due to the research, we all know that surface receptors, ACE2, which is a human angiostensin converting enzyme that is shedded in the lungs, arteries, heart, kidney, and intestines, has a strong binding affinity to COVID-19. Due to this, samples like saliva has been tested and is found to be a reliable tool to detect SARS-CoV-2. Besides that, saliva may also provide information about the clinical evolution of the disease, as it is very easily taken. So this sample can be taken on day 4, day 7, day 18, and at the same time, we can see the clinical evolution of the patient. A lot of papers done, for example, by Lorenzo et al., as well as Javed et al. From Iran, shows that the saliva gland that expresses ACE2 could represent a valid instrument for the COVID-19 detection. Besides saliva, stool is also another biomarker that is currently being actively researched. A small setback about the stool sample is that the PCR positivity still remains very low. But the plus point of stool is that it remains persistent throughout, as assessed by Wolf et al. As we can see, virus isolation from respiratory tracts, bronchoalveolar lavage, and sputum PCR are the gold standard. But at week 1, stool PCR can detect SARS-CoV. And this plays a very essential role in order to detect SARS-CoV among patients in that small window period of time. Besides that, 2 smaller studies have reported the presence of SARS-CoV in anal and rectal swabs. This presence can also show that fecal residue can or maybe transmit SARS-CoV. Several descriptive cases series have shown the presence of viral genetic material in the stool of SARS-CoV patients. This is a very interesting biomarker, tears. When it was first known that ACE2 is a receptor binding to SARS-CoV, and most coronavirus are oculotropic, viruses that bond to ocular surfaces of ACE2 receptors, tears came into a very big role to play. Unfortunately, sampling techniques, day of testing, day of collection has an impact on PCR results. Ocular tropism of respiratory viruses, for example, adeno, influenza, is a very well-known fact. That is why coronavirus in tears can be detected. Unfortunately, for samples like urine, as we all know that protein is expressed in the kidneys for ACE2, but there's a lot of conflicting data. However, 2 small studies done in China have published otherwise. A study by Yang Chang et al. Proved that urine pathopathology related to molecular alteration in urine is a potential application of diagnostic urinary proteinase, and the severity and determination as well as the development of COVID-19 can be done via urine. Proteins. In order to improve surveillance effort, tests using proteins are needed. Viral proteins are expressed, especially in serology methods. It is detected in the serum antibody of the S protein. Serum is an added value if it can be captured at the correct time of disease onset, because it can both capture the active as well as the past infection. Unfortunately, a lot of research show that IgM has a very high cross-reactivity. As such, it may not be able to play a very active role in case management. Other protein cellular markers. A lot of recent publications have published that other protein or cellular markers can be used for detection of COVID-19. A lot of papers have shown that patients have elevated D-dimer proteins as well as low lymphocytes, leukocytes, as well as blood testing. Patients with severe or fatal diseases have shown an increase or decrease in these markers. So predicting clinical worsening using this lab test can be used to aggressively see the progression or the severity of the disease. However, chest computed topography is also another new detection method that is being used. The only challenge, however, is using these biomarkers, they are also found abnormal in many other illnesses. As such, they cannot be used as a valid instrument for the detection of COVID-19. These are some of the references that I've had. Now, coming on to my second topic, the role of research laboratories during a pandemic, the COVID-19 experience. Here, I would like to share how is it that all the research laboratories came together in Malaysia during the pandemic in order to help out our Ministry of Health. We saw incredible dedication, collaboration, and innovation among the healthcare industries. Many organizations and teams came forward to overcome the challenges presented by COVID-19. Intertwined industry programs, relationship between the government, the NGOs, and the businesses played a crucial role in order to solve our social challenges at hand. In working with higher education as well as businesses that offer R&D, we saw how research laboratories played a big role. As we all know, many research laboratories in the public and the private universities provide healthcare facilities, and their lab most often have specialized and diagnostic testing. They also act as a reference lab. These unique capabilities proved very beneficial during this pandemic, as you know, as testing mounted, screening of people who were exposed, who were symptomatic as well as asymptomatic, was essential in containing the spread of COVID-19. As such, many of the healthcare laboratories could not cope with the high amount of testing. Hence, research laboratories came forth to assist the health ministry in its role to provide laboratory testing. We were very fortunate as we had many researchers who had the appropriate skills and testing principles and are well placed in the universities to carry out these tests. The assay to diagnose the novel coronavirus entailed the RNA extraction, the polymerase reaction, or the PCR. Now, these two methods were like second nature for most research laboratories and most pivoted into cholera testing centers within days. This was published in the newspaper, where we showed how many of our private as well as public universities came together to provide services on COVID-19 testing. Now, why is it that we were able to help out in a very short period of time? Unlike the simple organization structure in most national health laboratories, the research laboratories are composed of numerous individuals, and we have complex elements to help us pursue our thirst and knowledge for science and medicine. Of this, the single most important asset that we had was the knowledge and expertise of our laboratory employees. Research laboratories offered things like molecular genotyping, whole genome sequencing as part of the laboratory investigation to aid in the understanding of these viruses. Now, this was very important because when the scientists in China shared the genome sequencing, which was done by the team by Zero Rao, who uploaded the virus protein into the protein data bank, a lot of research laboratories came forward in order to sequence this genome typing. Molecular diagnostic like CRISPR as well as Sherlock was utilized. Besides that, most facilities are equipped to perform a lot of complex procedures. A particularly interesting role that laboratories played in this pandemic was the integration of molecular typing, especially in regards to epidemiologic information that helped us achieve true real-time epidemiologic surveillance. Surveillance typing network improved the outbreak detection, and this was made possible with the exchange of data between laboratories. Accurate laboratory-based information is a component for disease surveillance and is the highest goal in disease control. Hence, during a disease outbreak, samples should be sent to a research laboratory to confirm as well as monitor the characteristic of the organism. Notably, the willingness of scientists to rapidly share this information has enabled the rapid development of many assays, even before SARS-CoV started circulating outside of many countries, thus providing a timely and effective response. Another thing that the research laboratory in Malaysia did was, we were consulted, especially a few disease modelers were consulted in order for decision making. Now, these disease modelers actually assisted policymakers in making their decisions. This is one that was done by a team led by UKM, UNIMAS, and University Sabah. Besides that, University Malaya also developed a COVID-19 symptom monitoring system. UKM developed a COVID Sense app to conduct self-screening tests All these played a very essential role in the epidemiologic tracing. Besides that, researchers also went into simpler methods of testing for corona. They also tried to go into non-invasive procedure testing, also trying to find other biochemical markers or radiologic findings. Thus, research laboratory may represent the sole or the only center that has infrequently used diagnostic reagents. It is important that these unique facilities continue to be empowered not only to maintain their testing, but also be the supplier of reagent which there is limited commercial supply. As there was a lot of disrupted reagents coming into the countries, a lot of basic items like flock swabs, VTM couldn't be received by a lot of laboratories that were testing in the Ministry of Health. We were very lucky because we had our own media preparation facility during this pandemic. So we could prepare our own VTMs. And because we had a very good networking with a lot of NGOs, there was a center that was doing cervical testing for human papilloma viruses. And we got flock swabs from them, about 3,000, to tide us over before our next supply came over. This is a picture of our VTM testing, where instead of using plastic bottles, we reverted to glass bottles that could be autoclaved and cleared after that. A lot of centers also did their own PCR extraction, PCR machines, as well as swabs. Besides that, research centers also had autoclaves, state-of-the-art 3D fabrication machines. All this came into play in helping the local government and healthcare who had problem responding with physical needs. A team from the Computer and Science Faculty and Information Technology UNIMAS printed face shields for healthcare workers. They also made ear guards to help protect healthcare workers during these times. This is a picture of the face mask that they did, and this was made specifically to the specifications set by the Ministry of Health. Besides that, a lot of universities came up with their own hand rub solution. Some, they also did it with supplies that can be found around us in the environment. Besides that, healthcare workers were also helped by the Faculty of Engineering, who made intubation boxes. This is a box that is a transparent tube that covers the patient's head during endotracheal intubation period procedures. Besides testing, research universities also came up with treatment and vaccine. The University Technology Mara pooled their resources by combining their diagnostic machines as well as working with Pharmacogenetics Institute to help set up a database to identify which genes could be used to develop tests. University Malaya led clinical trials for the COVID-19 treatment. Around the world, the Pasteur Institute, a French research institute that studies diseases and vaccine, joined together with the University of Pittsburgh to lead a global networking with other universities, biotech firms, to test drugs that could be useful for treating COVID-19 while changing the way the world does science together. Besides testing, vaccine, universities also played a very crucial role in offering technical guidance. Before I end, my take-home message is, no one organization has all the resources, all the expertise, with all the scientific and medical complexities that could handle if ever another pandemic were to be present. The ability to work collaboratively is very important. To date, researchers have identified and shared hundreds of papers and more than 200 clinical trials, bringing research laboratories, diagnostic laboratories around the globe together. Effective communication among all key players are a very crucial aspect that should be enhanced during any pandemic. Thank you very much. Any inquiries, please email harvinder.training@gmail.com.
Thank you very much, Harvinder, for an interesting and comprehensive presentation on how numerous institutions get together for the common good of combating the coronavirus. Now, before we proceed to the next talk, I wish to remind that the participants can actually start posting your questions anytime during the session on the Q&A chat box, on the Q&A box. Our next speaker is Mr. Tin Tun. Tin Tun, can you show your slide? Tin Tun was trained as a medical technologist in Myanmar. From there, he moved to Singapore for work, specifically in the virus lab in SGH, Singapore General Hospital. While he was there, he obtained his MSc from the National University of Singapore. All this while, his interest was focused on biosafety management and workplace safety and health. He is an IBFA certified professional, and that is the International Federation of Biosafety Association, Canada, and also a registered biosafety professional with the American Biological Safety Association. In Singapore, he is a Ministry of Manpower's approved biosafety coordinator and risk consultant in workplace safety and health, as well as an accredited trainer and assessor. Professionally, he has served on the board of AACLS and SAMLS, that is our Singapore counterpart. His last position before retirement was Biosafety Program Manager, Nanyang Technological University, Singapore. Tin Tun, you can take over.
Okay. Very good morning, everyone. First of all, I would like to express my appreciation to the organizing committee, MIMLS, Malaysia Institute of Medical Laboratory Science, and AACLS, the ASEAN Association of Clinical Laboratory Science, for giving me opportunity to share my view at the seminar. I will be talking about Biosafety Level 2, BSL2 laboratory, biosafety requirement for the testing of sample associated with the sample associated with the COVID-19. But I am not sharing the how to test the sample to test the COVID-19. The one already, Ms. Aung have already shared my review. So for my experience, actually in microbiology, my background is microbiology in Myanmar. Then I moved to Singapore and then virology. And at Nanyang Technological University, I look after BSS3 laboratory and then biosafety, university biosafety program. Now, the world is facing with a new virus, novel coronavirus. Novel coronavirus. The originating from the Wuhan, China. And then WHO declared PHEIC, Public Health Emergency of International Concern, on 30th January 2020, and then pandemic on 11th of March to 2020. That the impact of the COVID-19 is a lot. The 213 plus countries are affected. And more than 5.6 million of the confirmed cases are the infected, and the 356,000 with the death. The impact is a lot. So the world is busy with the COVID-19. Every country is trying to respond to stop to prevent spread of the disease. There are so many strategies: circuit breaker, lockdown, and the quarantine, the work from home, and then closure of school and business, and etc. And social welfare, billions and billions of dollars are spending for the social matter, and then the jobless issues, and then the resettlement grant, etc. And then public health awareness, personal health awareness, stay distancing, home stay, mask wearing, and then hand wash, etc. For patient care is a hospitalization, ICU, and then treatment regime. We are looking for the treatment regime also. Public health concern, yeah, a lot of country or research group, they are trying to produce a vaccine for prevention, that contact tracing, and then mass testing, and then laboratory testing. So the whole world is busy with all these things. And then before they were testing the COVID-19, we have to conduct the risk assessment, RA. RA says that there are three parts. Number one is to identify the risk associated with the COVID-19. And then number two is evaluate the risk. And number three is for the control measures to prevent the LAI. The COVID-19 is highly infectious, and mild to severe, and then what we call cough, a fever, and the pneumonia. The transmission is also contact, the direct contact, aerosol contact, etc. And then stability of virus also important. Therapeutic renewing is at the moment we don't know. That's why we have to find out the new one. Evaluation, yes. According to the risk level, the COVID-19 is assigned to the risk group, high risk group number 3. In Singapore, we call it the schedule number 1, likely of causing the LEI, handling sample, live virus or inactivated virus, a facility requirement. We have to all consider safety procedure. We have to all consider. And then for control measures, we have to use the good microbiological practices, SOPs, as a minimized social aerosolization. We have to wear standard PPE and N95 masks, and then frequent hand wash, etc. I'm sharing the two graphs. This is for Association of Public Health Laboratory. This from their study, the patient 1, the 4 days after onset of the disease, and the viral load is detected. And 5 and 6 days, and then they are going to find out the highest viral load there. And then 8 to 9 days later, and then virus is not detected. And then for patient 2, it's a bit earlier, 3 days after onset of the disease, virus was detected. And then 5 and 6 days, the highest viral load. And then after that, 10 to 12 days. And then after 10 to 12 days, cannot detect that. So their study, they show us when we should test, trying to detect the virus in the sample of infected person. So this assessment is going to provide the very, very important information. Number 1, selection of the appropriate biosafety level, at which level we are going to use. And number 2, microbiological practices and procedure. Yeah, procedures we have to recommend. And then use of the safety equipment, facility requirements, and that can prevent the LAI, laboratory acquired infection. So regarding the biosafety level defines the biosafety status of the facility. There are the 4 status, BSL 1, 2, and 3, and 4. 1 is for low risk to handle low risk, 2 is for moderate risk, and then 3 is for high risk and risk group 3, and then 4 is the highest one, therefore research purpose. So, conducting the RA, who is responsible? Is the laboratory director, the principal investigators of the research group leaders, the IMBC, the Institution of Biosafety Committee and other committee, and the biosafety professional and lab users are responsible to conduct RA. So Ministry of Health in Singapore, and they recommend, because biosafety requirements depends on which method you are going to use, which method you are going to perform. For example, serological assay to identify an antibody, antigen detection, or maybe molecular assay, PCR, sequencing, genetic manipulation, and the pathological examination of the fixed tissues. These methods, if you are going to perform these methods, what are the biosafety requirements? There's a BSA2 laboratory is required with enhanced biosafety practices. That means we can call it a BSL2 plus is required. In Singapore, we also call it uncertified facilities. But if we are going to culture or isolate handling the live virus, yes, BSL3 facility is required. This is we call it certified facility is required, and the BSL3 procedures and practices are must be applied. So sample testing the sample of the clinical sample for COVID-19. We use a nasal swab and throat swab, a sputum, and blood, and other samples. But in common, to transport of the sample is also important. Then WHO, once triple packaging is we have to apply. PCR testing, yes. PCR testing is at the moment, the PCR, many countries, many laboratories, this is method of choice. The PCR is actually the tell presence of virus in person or in patient, in sample. But no, PCR doesn't tell how and when infected. But PCR does have the advantages. Its sensitivity is high, and the selectivity and accuracy and producibility, because that's why one of the method of choice. And that what have the challenges, a big ways, serological assay to detect viral infection, past viral infection, antibodies that produce to fight the virus. These serology assays are not broadly validated for clinical use at the moment. So BSL2 facility, BSL2 laboratory facility requirements, basically the laboratory should have negatively pressurized. The idea is to contain all the risk inside, never release out. And biohazard signs should be there. And then laboratory is a not a place to easily assess. The strict access control should be in place. And the biosafety cabinet should be equipped with the biosafety cabinet. And then all the safety equipment like autoclave and centrifuge and whatever, they are also well maintained and keep functioning. A biosafety training should be provided for all the lab users, and the biosafety manual must be developed, and the RA, yes, risk assessment must be conducted before covering all the activities in the laboratory. And then handwash facility have been provided. These are facility requirements to test for the BSL2. And the enhanced procedures and practices, basically good microbiological practices must be applied. And then we trying to minimize aerosol to stop the LAI. Then all the procedures that for handling COVID-19 sample should be done inside the certified BSE. And then all the lab users, the standard PPE, like at the N95 mark, they have to apply a sharp control in the lab. It also must be in place. A bio-waste management is also one of the important things. All the generated waste should be treated very well. And then in case of emergency preparedness, yeah, also important as peer management and incident reporting, etc. And medical surveillance, fever screening, temperature screening, symptom screening, all have to be prepared. And then the inventory is also biosecurity breach. So inventory record must be applied in the lab. So ultimate safety depends on the how you work. And then because in laboratory got many people, and then they all have to aware of the safety and then good teamwork, rather than how good facility is or what you know or SOP. If you don't apply them, it's not very little use. So it depends, total safety depends on how you work. Then there's a protective clothing, then you have to wear the lab coat and disposable gown and then cover all. Eye and face and eye protection, you got the goggle and face shield, etc. And then there's respiratory protection with the mask and the surgical mask and then N95 masks. All are single use, disposable respirator. Surgical mask is actually designed to protect patient, not for the wearer or not for the laboratory user. But N95 mask, yeah, better protection for the user. The N95 mask test for the mark fitness test should be done by the qualifying person and full protection. So due to the high demand during the COVID-19 period, and then there's a global shortage of the mask is that when we have the facing with the problem. So some laboratory like hospital infection, they trying to make surgical mask or reusable using the autoclave 121 degrees Celsius for 15 minutes. And then NIH, National Health Institute, NIH, they trying to make reusable the N95 masks using the 4 methods: PHP, vaporized hydrogen peroxide method, dry heat method, 70 degrees centigrade, and the UV light and 70% alcohol spray. They publish all these methods are working. So hand protection to handle infectious and the blood or body fluid or whatever, all have to we have to wear the gloves and disposable one, microbiologically approved latex, vinyl, nitride gloves and use. And after using, we have to dispose as the bio waste. Then some get allergy, and then dermatitis, etc. So powerless gloves should be available. And then for reuse, not recommended for the gloves. Then hand protection and how to remove the gloves. How to remove the glove without contaminating, because outer surface of glove is contaminated. So when you remove, try not to touch outer surface step by step, and then after taking out, go into the biohazard bag, dispose the biohazard bag. And then MOH, they recommend a step of the cleaning your hands, the palm and the fingers, back of hand, and the base of thumb, or step by step, and it takes about 20 seconds for the proper hand wash. So Singapore is working hard to get the green environment, and then Singapore the green city. Any inquiry, please email to intern64@gmail.com. Thank you very much.
Thank you, Tinton, for your comprehensive coverage on the laboratory infrastructure required and measures taken for the testing of coronavirus. Before we proceed to the next speaker, I would like to remind that questions for the speaker should be posted in the Q&A box, and in the chat box will not be entertained. I repeat, post your questions in the Q&A box and not in the chat box. Our next speaker is Dr. Zainul. She's a graduate from the University of Surrey, UK, where she obtained her BSc Honours in Biochemistry. She worked there as a RA for a year and had to return to Brunei to serve in the clinical chemistry laboratory in Repas Hospital. Her interest in biochemistry waned when she did her Master of Biomedical Sciences at the Curtin University of Technology, Australia. Returning from there, she had her eyes fixed on virology. Dr. Zainul is now attached to the Virology Laboratory of Sembiling Biomedical Sciences Research Unit, Ministry of Health, Brunei. To complete her academic achievement, she is now a proud PhD from the University of Sydney, Australia. Dr. Zainul, please.
Thank you very much, Mr. Woon. Assalamualaikum and a very good morning to all of you. So firstly, I would like to thank AACLS and MIMLS for inviting me to speak today. So today I will be sharing with you our experience in managing the surge during the COVID-19 outbreak. So probably a little bit of introduction on laboratories' response to an outbreak. So one of the pillars of an effective response to an outbreak is an efficient laboratory system. So according to WHO, the timeliness and accurateness of the result can be threatened when testing demands exceed the capacity of testing. So this happens when there is a backlog for testing and it is no longer possible to turn around results within 24 to 48 hours. The demand for laboratory reagents exceeds the capacity for supply. Laboratory staff are exhausted and working hours need to be reduced. The number of incoming samples exceed the capacity for safe pre-testing storage. Critical staff become infected or otherwise unable to perform their duties. Example, they are being quarantined. As well as laboratory instruments that can no longer be serviced or properly maintained. So all of this can actually affect the timeliness and accurateness of laboratory result. So unlike Malaysia and other countries, in Brunei Darussalam, only one laboratory are designated to conduct COVID-19 diagnostic testing. That is the National Virology Reference Laboratory. So this laboratory also conduct other routine virology tests, serving all community and public health centers and hospitals in the country. So this laboratory conduct routine serology tests as well as molecular tests, and staff are rotated on a monthly rotation basis. So prior to the outbreak, the laboratory has a capacity to run approximately 60 samples per day for molecular testing only. So this photo is to show you the building facility that we are currently in. So the photo on the left is the photo of the laboratory building located in the capital. So molecular labs are located on the 3rd floor, and it is a biosafety level 2 laboratory with enhanced BSL2 practices. So the photo on the right is just showing our virology staff team taken during our ISO 151 assessment sometime last year. So in terms of SARS-CoV-2 PCR tests, Brunei started testing on the 24th of January 2020. So we initially uses a commercial PCR kit detecting the open reading frame 1A and B for our primary detection, and further on to expand to detect E gene and RDRP gene based on the protocol by Corman et al. So we did the extraction manually using the QIAGEN RNA mini kit. So this is to show the summary of our detection method. We detect the open reading frame 1AB using real-time PCR commercial kit produced by BGI. So this kit was used as our primary screening method. We also use E-gene and RDRP real-time PCR manufactured by Tipmol Bio So usually we use these kits for confirmation, or when we are actually we have doubts in our screening method. So this is our workflow for our molecular testing for COVID-19. So upon receiving samples, we sort and register sample into our laboratory information system, which we call BRUHIMS. We then do nucleic acid extraction, followed by master mix preparation, and then loading the nucleic acid onto the master mix, followed by detection and result analysis, and finally, result entry and release into our laboratory information system. So during this peacetime, only 2 staff tasked to perform all these steps. So on March the 9th, 2020, Brunei reported first positive COVID case. This case is a 53-year-old male who had returned from a Muslim religious gathering in Kuala Lumpur, Malaysia on the 3rd of March. So during the first 3 weeks, there was a rapid increasing number of contact tracing testing that was sent to our lab. So this chart shows our daily sample workload in yellow and our positive sample in the red bar. So you can see our sample workload reached to more than 500 per day during the first 3 weeks of the outbreak. So we receive samples for anyone with any contact history, regardless of symptoms, anyone with travel history, regardless of symptoms, all pneumonia, any secondary presentation for influenza-like illness within 14 days. So additionally, surveillance was also done in foreign worker, which started sometime in May 2020. So we faced lots of challenges during the outbreak. So our laboratory ability to adapt was limited by the number of suitably trained and experienced staff who could be brought in to provide the search assistance. Staffing changes in roles and responsibility have also impacted our workflow and processes that have required the lab to adapt quickly to manage the search. We also encountered supply issues during the first month of our of the outbreak. We experienced shortage in for example, viral transport media, as well as N95 mask. So it made it difficult to manage during this difficult time. So how do we manage the search during the outbreak? So here are some of the things that I've listed, which I will describe further in the next few slides. So the Department of Laboratory Services activated an emergency operation center as part of the laboratory preparedness plan. So the function of this emergency operation center is where all members consisting of heads of clinical laboratories meet and discuss responses to an outbreak. So assessment and immediate response was made to support the laboratory needs in terms of the staffing, the cross coverage of technical staff and training needs, the equipments, scaling down of non-critical laboratory tests, reporting, as well as logistics. So as everybody know, high level of expertise are required to perform molecular testing. So so there was another issue that need to be considered when we manage the search. So during the initial outbreak, all our routine technical personnel within the laboratory are mobilized to do the SARS-CoV PCR testing. There was also deployment of technical personnel from other laboratory to cover the PCR testing, as well as for other our routine virology tests. So team of staff was also identified with specific roles and responsibilities for the different stage of PCR process. So we actually identified 3 staff per team for each of the working shift. So for the deployed personnel, the training had to be done quickly. So we did this by allowing them to work shadow the experienced staff and slowly taking over the task once they are competent. Coordination with other clinical laboratories in providing support for the pre-analytical processes was also very important. So, so in this case, we do not need an expert staff to actually handle. So this is just the pre-analytical processes where where the samples need to be labeled, sorted, and registered into BRUHIMS. So we other laboratories can help this very easily. So we also ensure our staff take the obligatory off-duty rotation to ensure that they are well rested as much as possible. So before the outbreak, we had to we test SARS-CoV-2 only during office hours, 7:45 until 4:30 p.m. But we have also staff on call after hours and after office hours and weekend in cases of urgent specimen. However, during the outbreak, we had to extend our working hours in order to manage the sample search. So we actually extended our service from 7:45 until 4 a.m. on a that is on a rotation basis, 2 shifts per day. So we also manage our sample search by optimizing our workflow. So with the increasing sample numbers, the PCR testing was implemented into our routine laboratory workflow that that has already exists for the diagnostic testing of other pathogens. So staff are already familiar, and this will minimize the disruptions. Gradual increase in sample batch processing was also done once the staff feels more confident in handling large number of samples. We also have dedicated staff for each PCR processes during each work shift to minimize cases of contamination and repeats of the PCR reactions. We also optimize our workflow by using barcoded work lists for ease of test ordering as well as entering of result into our laboratory information system. So during the first week of the COVID-19 outbreak, we experienced a very high sample backlog. So we actually had to refer our specimens to National Public Health Laboratory Singapore. So they have actually kindly helped us analyze 500 samples to clear our backlogs during the early outbreak. So samples were sent via World Courier as a category B shipment. So I think leadership and tasking are very important during the difficult times. So I must acknowledge the leadership in the Department of Laboratory Services during the outbreak. So it is important to have as much support to manage the laboratory search. So in our case, we have identified a number of people to look into various matters. For example, management and testing, which include workforce management, would be the head of the laboratory. In terms of search response, there is a team of people in the Emergency Operations Center where they will actually respond and deploy staff when needed. So daily reporting of test volumes and result required by the Ministry of Health daily would be handled by the Emergency Operations Center secretariats. So we also have people tasked to look after our laboratory and equipment supply, as well as to look after our PPE supplies. So by the consent of His Majesty, Brunei established an extension building of the virology lab in an effort to meet the rising demand of the COVID-19 testing. So the building construction completed within 2 weeks and opened on the 30th of March 2020. So in this new facility, there are segregated areas for different stages of PCR processes. It is also equipped with automated nucleic acid extraction system. So our testing capacity, we managed to increase the testing capacity 10 times as compared to previous capacity. So this is the photo of the new extension laboratory, which is just next to the existing laboratory, which I showed earlier. So this is showing the interior of the lab with the different areas for the PCR processes. So, so as of yesterday, Brunei recorded a total number of 141 cases. There are no new cases for the past 28 days. There are 138 recovered cases, one case still active, 2 deaths, and as of yesterday, since January 2020, our laboratory has conducted 20,619 tests. So in summary, the ability of laboratory to deploy PCR testing was felt to be a key for successful response to an infectious disease outbreak. As such, testing is highly sensitive and specific. So we believe that there will be future infections outbreak that will strain the standing capacity of clinical laboratories, requiring effective implementation of such capacity response. So that ends my presentation. Probably this is the best time to acknowledge the unseen heroes fighting the global pandemic, that is our laboratory professionals. Thank you very much, and I can be contacted at zainun.zaini@moh.gov.bn.
Thank you very much, Dr. Zainun, for walking us through your difficult time. I do not envy your life during this pandemic. I also empathize with your stress in handling the search, especially with the very lean staff of 6. Thank you. Our next speaker is Mr. Christian Bujak. He is from Germany and has a master's degree in biomedical engineering from the Technical University of Munich. He wrote his diploma thesis at the École Polytechnique in Paris, France, where he investigated the flow response of endothelial cells in the context of development of arteriosclerosis. He has spent some time at the MITI laboratory, and that is the minimally invasive interdisciplinary therapeutical intervention at University Hospital at the Technical University of Munich, Germany. At the MITI laboratory, he was responsible for the enhancement of automated surgery process. In 2015, he joined Draeger in Singapore, has now risen to Regional Marketing Manager, Respiratory Care and Neonatal Care for Africa, Asia, and Australia. Mr. Christian, the floor is yours.
Dear Mr. Woon, thank you very much for this warm introduction. Dear Chair, dear committee, and of course, dear fellow speakers and dear attendees, thank you very much for taking the time. Today, I would like to talk to you about jaundice screening during COVID-19. Terima kasih from my side. So I would like to give you a quick overview of the agenda. Before we talk about jaundice screening during COVID-19, we're going to talk a little bit about the physiological background of jaundice and current screening measures. And then we will talk about efficiency improvements that have been made and that can especially help during COVID-19. So also a big thank you to be part of this shared experience. It's a different kind of lab testing. Of course, the topic of COVID-19 remains, but in this example, we will be talking now about jaundice screening. Also, thank you to the AACLS and to MIMLS as the organizers of this event. So let's start with what is newborn jaundice? What are those factors? In fact, newborn or neonatal jaundice is very common. We can see here on the right, we can see an example for for a newborn with jaundice. And in a full-term infant, usually jaundice is apparent within 2 to 4 days after birth. Now, how does jaundice develop? As all of you know, of course, it highly involves the molecule of bilirubin. So I wanted to give you a quick overview of the bilirubin metabolism, showing the different steps. So as we can see in the upper left corner, we start with the red blood cell. And of course, once the red blood cell is breaking down, we have the hemoglobin molecule. And then as part of this, the heme molecule. And further down, it will go transformation from biliverdin, then attaching to the albumin protein, transferred to the liver, where actually the molecule is being processed and conjugated. And then finally, we see the excretion through the intestines. So these are the different steps. We want to, of course, differentiate between indirect or unconjugated bilirubin, which we see in the stages number 1 and 2, and then afterwards we have the conjugated or direct bilirubin. Here I also wanted to include some of the usual lab testing methods. I mean, the original reaction was the Van Berg reaction. Today there are also different reactions which can be used to measure bilirubin concentrations that you can see down in the right corner. Now, why is neonatal jaundice so common in newborns? In fact, we see that the bilirubin load of a newborn is 2 to 3 times the amount of an adult. So out of the blood, we see in fact that up to 85% of the produced bilirubin is coming from the breakdown of red blood cells. So once we then see at high bilirubin levels, we see the typical yellowness of the skin because bilirubin is deposited in the skin. This is, of course, what makes it so visible. The pathophysiology, in fact, is that we see an imbalance between bilirubin production and bilirubin clearance. So this imbalance can have several different causes. In the newborn, we can actually talk about a physiological jaundice. This is something which is very common. And usually this jaundice is being treated with either phototherapy in mild to moderate cases, or can also be treated with a blood transfusion in very severe cases. However, if this high concentration of bilirubin, so the hyperbilirubinemia, is not treated, this can actually lead to severe physical and brain damage, so which then would be called Kern icterus. Now here we can see a quick explanation of Kern icterus. As we know, the unconjugated free bilirubin is fat soluble and can therefore cross the blood-brain barrier and can settle around brain cells. In the beginning, I mean, it might only temporarily affect brain function and is therefore reversible. However, over time, this impairment can get more severe and might cause permanent brain damage. And this permanent brain damage is then called Kern icterus. And we can see here in those pictures, we see really the consequence of Kern icterus. This can lead to cerebral palsy, hearing loss, upward gaze, or also defects of the dental development. Unfortunately, there is no cure for Kern icterus. So once the hyperbilirubinemia has developed to a Kern icterus, this cannot be treated. So therefore, we need to be vigilant, monitor the newborns closely, treat them appropriately, and of course, make sure with the right follow-up that there are no long-term consequences. As mentioned before, in fact, 2 out of 3 newborns experience signs of jaundice. So this is, of course, a very huge part. And for preterm infants, so infants which are born before their usual gestational age, we can actually see that it's more than 90% of those preterm infants which are showing jaundice. Now, what is the crux here? I mean, yes, we see that these high bilirubin levels are more or less physiological, but when we look at the typical time a newborn spends in the hospital, we can actually see that usually the newborn is being discharged from the hospital within, let's say, the first 60 hours of life. However, when we look at the bilirubin levels within the blood, in fact, those levels are increasing a lot later. So another 24 to 72 hours later, depending on when the child has been discharged. So therefore, there's a big risk of missing a case of hyperbilirubinemia. Now that we are aware of the side effects or of the potential impairments of high bilirubin levels, how do we screen for jaundice? Now, newborn jaundice can be assessed in these three different methods. First of all, we have the visual assessment where we have color maps and looking at a color comparison of the skin. Then we have the universal total serum bilirubin testing, which is done with the heel prick and in the lab. And a rather new development is the universal transcutaneous bilirubin testing. And we're going to talk about these two methods in more detail. So, as you all are, very familiar with the TSB measurement, just quickly talking about some of the advantages and disadvantages of this method. Of course, it's the gold standard. It's clearly the best method to determine accurate levels of bilirubin within the serum. And of course, we can also measure both unconjugated and conjugated bilirubin, so we can really assess the pathophysiology of the infant a lot better. However, as you can see in this picture down here, it does require a rather painful procedure, this heel stick draw, which, as you can see in the picture, is not very comfortable for the newborn. And as we see also from the earlier slide, it may be too early to determine the accurate levels. So maybe at a later or a second time point, a second heel stick would be necessary. So therefore, these are some of the disadvantages of TSB. When determining the concentration, of course, it's then to assess whether or not this is a bilirubin level which requires further treatment. And to assess this, usually nomograms are being used. And here you can see Dr. Bhutani's nomogram. So we see the postnatal age for a term infant. So you see this in hours, and of course on the y-axis the serum bilirubin levels, and you can see then in here, based on this nomogram, the different risk zones, which infant is at risk. So for instance, if we see a measurement of a TSB of 10 milligram per deciliter after 46 hours, we see actually we are in between the high to low intermediate risk zone. This is how it's been being read and interpreted. And then this is one part of deciding the next steps for this infant. Of course, the second part is a close look at risk factors. So we always need to combine the actual TSB reading and take into account further risk factors. Here's just an overview of some of the major and minor risk factors. So for instance, if jaundice is already visible in the first 24 hours, this is a good indicator that we are looking at a rather severe case. Also, low gestational age is another potential indicator, and you can see down here further risk factors. So, how can we minimize the trauma for the newborn while actually increasing the screening? Because I mean, we saw the potential side effects of high bilirubin levels. So we want to look at a non-invasive measurement, and this is the total transcutaneous bilirubin measurement. You can see this right here, either measured at the sternum or at the forehead. It's very easy to use, a very quick way to test the transcutaneous bilirubin levels, can be easily repeated and allows a very close monitoring of the infant. However, and this is very important, there are some things to keep in mind. When implementing a TCB measurement, it's important to develop a site-specific protocol. At which TCB level would you then follow up with a TSB measurement? This is one important thing to state. A TCB cannot and will not replace TSB. The main purpose is to reduce the amount of potentially unnecessary TSB measurements. So therefore, we want to monitor the development of the newborn and then define a threshold at which we want to confirm our suspicion or confirm the diagnosis with a TSB. So this is the average workflow. For TCB measurements, there are different nomograms. This is important to state because sometimes there's a mix-up between the different nomograms. When we are looking at transcutaneous bilirubin levels, it's important to use the Masel's nomogram. So in this case, when we look again, actually the same measurement of 10 milligram per deciliter at 46 hours of life, we are here. We are at a different point, at a different percentile for this nomogram. So it's important to choose the right nomogram for the right measurement to then determine the next steps. So as you can see, once again, on the left, the nomogram from Dr. Bhutani for TSB, on the right, Dr. Masel nomogram for TCB. It is, however, possible to use the Bhutani nomogram as well, but then it's important to use the 75th percentile. So this one right here instead of the 95th percentile. Now, what is the measurement principle? So the JM105 uses optical measurement. So we have a light emitting path in the middle, and then which is basically the sender of the light signal. And then we have a short optical path and a long optical path. So the measurement principle is actually quite simple. We are sending out blue and green wavelengths. And by using these two optical paths, we are measuring the yellowness of the skin. And we can also take into account the different impact of the skin. So we can see here very simply the blue and the green spectrum, and the more bilirubin we have in the skin, the more yellow the skin is and the more the blue light will be absorbed. The performance specifications you can see right here, the measurement range going from 0 to 20 milligram per deciliter. Or of course, if you want to measure in micromole, it's 0 to 340 micromole. The accuracy depends on the gestational age, because we see that for preterm infants, the accuracy is slightly decreased. And this is a rather new development. The device can also be used after the initiation of phototherapy with another additional decrease in accuracy. So for the term baby, it's plus minus 1.5, and then can go up to 2.3 milligram per deciliter. One important step when performing a measurement is, of course, to make sure that the measurement will be accurate, that the test equipment is still well calibrated and the measurements and the readings can be trusted. This is why the JM105 has an included light source checker, which can be used before the measurement. You can actually see the reading right here to make sure that the wavelength and the measurement are within specifications. So how would you then do such a measurement? How would you take a measurement? I mean, we need to clean the probe, then place the probe onto the skin in a perpendicular fashion. It's important that there's no air or nothing between the actual probe and the skin. And as I said before, it can either be measured on the sternum or on the forehead. Those two measurement sites, just as a general information, both can be used in usual inpatient setting. However, when you're looking at an outpatient setting, so if the infant has already left the hospital, we only recommend to use the measurement at the sternum, because now once the child has been exposed to sunlight or daylight, the reading on the forehead cannot be used as accurately as before. And depending on the measurement protocol on site, you can either just take one spot measurement, or you can actually have up to 5 measurements and simply save the average of those 5 measurements. So this is the overall workflow. We are screening the newborn transcutaneously for the progression and development of bilirubin levels. Once it's past a certain threshold, which we are determining with the right nomogram, we are then performing a TSB measurement. This TSB measurement, if this confirms the suspicion, then you can start phototherapy, which you can see in the upper right corner, and continue phototherapy. Of course, after the initiation of phototherapy, it's important to continuously monitor the bilirubin levels in the infant, because you need to make a decision when to stop the phototherapy. So this can be monitored with TCB, but again, the decision to stop phototherapy needs also be confirmed with a TSB measurement. Now, just a quick overview. I mean, TCB measurement has been already introduced a couple of years ago, and I mean, in general, the respective neonatal care societies have already included this in their neonatal jaundice management guidelines. Here, I would like to share with you the guidelines from Malaysia. You can actually see in the upper part right here of the recommendation that all babies should be assessed at every opportunity. And you can already see the different thresholds at which a total serum measurement should be done. So this is just a quick excerpt of the guidelines from Malaysia. Of course, here's another guideline, for instance, from Australia, where you can also see a flow chart, including also some of the limitations or contraindications for use. And of course, really stating the fact that each site needs to implement their own protocol. Of course, they can use predefined protocols, but at the end, it's really important to make this site-specific. One open topic in literature is still the fact when to stop phototherapy. Here I just shared 3 different publications which show that actually there's no standard for discontinuation of phototherapy. One suggestion might be at a level of around 13 to 14 milligram per deciliter to discontinue phototherapy, or in this case, in this other publication, 50 micromole per liter below the initial phototherapy threshold. So as you can see, this is still not clearly decided, and some further research needs to be done in this field. So we were talking about the efficiency improvements, and we are slowly going to talk more about the role of TCB during COVID-19. So in this study from the year 2011, Weiner has looked at almost 15,000 healthy infants and compared the workflow before and after the introduction of TCB measurement. And one very promising result was, in fact, that by using TCB measurement, in fact, there was a 54% reduction in the incidence of very high total serum bilirubin values. So by increasing the monitoring, it could be prevented to have these rather severe levels. On top of that, there was a significant reduction of TSB draws down from 134 to 103 per 1,000 live births. So this is already one promising result of not just saving costs, but of course also improving the potential outcome for the newborn. In this study from the year 2008, McLean also looked at cost savings. So after looking at the introduction of TCB, they were able to decrease the amount of TSB by 71.4%, while at the same time increasing the number of screens from 1,300 to actually 2,700 per 1,000 live births. A second positive effect, and we will come to this even more, is of course the amount of time necessary for a screening has been decreased from 12 minutes down to less than 3 minutes. So of course this also comes together with significant cost savings. And lastly, I just wanted to share this. There are still some open topics in transcutaneous measurement, and this is of course transcutaneous bilirubinometry in preterm infants. I mean, I did mention this already shortly, and also in infants after the initiation of phototherapy. In this study right here from the Netherlands, they could also see that blood sampling could be reduced by 42% with only 2% of false negatives. So another very promising result. So with this TCB measurement, we can see already certain efficiency improvements. Now, how can we apply this in to the COVID-19 settings? And I wanted to share some very creative solutions. So during COVID-19, I mean, there were many articles, of course, also looking into COVID-19 and how it affects children. And in general, the overwhelming evidence points to the fact that children in general show milder cases and a better prognosis than adults. But nevertheless, I mean, as we all know, children are not immune to the disease. They might still get sick. And if you see in the conclusion, yes, they had a better prognosis and deaths were extremely rare. But still, I mean, I think many of us are parents. And of course, I mean, it's very important to save every single newborn. So therefore, we need to make sure to also apply the right precaution measures when screening newborns. How are newborns infected? This is another publication from the CDC in the US. They looked at the route of transmission for newborns, and they saw that they primarily were infected through respiratory droplets during the postnatal period when the newborn was exposed to either mothers or caregivers, visitors, or healthcare personnel with COVID-19. So therefore, this is the main route of transmission for those newborns, and tells us already some things to keep in mind when we want to protect the weakest of them all. So should we now just stay at home and and not screen newborns? I mean, of course, this cannot be done. I mean, we need to find a way to live with the virus and to find the right measures. So this is another recommendation from Babies First Test. This is another US government site looking at the different screening methods and looking at screening for newborns during COVID-19. And a clear recommendation is that babies should still receive newborn screening, but we need to make sure that the facilities are taking extra precautionary measures to minimize the exposure to COVID-19. So with all this combined, we want to protect parents, newborns, and caregivers alike. We want to minimize the risk of infection by minimizing the exposure. I mean, we all know about, work from home, stay home, social distancing. We have had, a very good talk already on the importance of PPE and having the right protective equipment. So how can we now take the jaundice screening and move it rather to the outpatient setting? Because of course, if we are outside of the hospital, then of course we are minimizing the exposure for everyone involved. So in the outpatient setting, usually we see neonates who have been discharged from this intermediate risk zone of the nomogram that I talked to you about just previously. And those neonates are usually seen within 72 hours after discharge from the hospital. So also there, the workflow is as follows. If then the TCB level is too high, a serum bilirubin needs to be drawn to then decide whether or not to initiate phototherapy. So what can be done? I mean, in the outpatient setting, and we saw, of course, many creative solutions already for the COVID-19 testing. So we talked about swab testing and different methods. So what we saw, of course, now it was first in Korea and in the US, we saw the initiation of these drive-through testing sites for COVID-19. Just very recently, there was also a testing site established in Singapore. This is from CNA News on June 3rd. So this is actually a very recent development. So with all this combined, I'm very happy to share this very creative idea coming from Malaysia. This right here is actually a Facebook post of the Director General of the MOH of Malaysia, who shared this experience. This is, of course, what also the whole talk series is about, experience sharing during COVID-19. And this is a very creative solution. To be honest, this is the first time I've seen this almost across the globe. So I was very happy to see at the HRPB hospital in Ipoh, there was a drive-through jaundice screening was implemented. It was used to prevent crowding, decrease the turnaround time, and make sure that social distancing can be done. On one day alone, they screened 63 babies. And out of those 63 babies, only 11 babies required a venesection, and 3 babies required eventually phototherapy. So this was so promising that the team in HPRB will actually continue this practice even after COVID-19 has passed, because it's simply such a very efficient process. You actually see right here, this is a newborn sitting in a car seat. You can see this right here. And as mentioned before, in an outpatient setting, the correct measurement spot would be the sternum of the newborn. So a very creative solution, which I wanted to share with all of you. So to sum it up, of course, we want to prevent the baby from kernicterus and protect the brain of the child. We want to also reduce pain and stress for the baby. So therefore, the combination of TCB with TSB can be really highly efficient, especially in the outpatient setting. This way, we can actually increase the number of screened newborns, reduce incidence of severe bilirubin levels, reduce screening time, and allow this fast screening, help save costs, and at the end, of course, we're minimizing the exposure to COVID-19. And with this, I would really like to thank you all. Terima kasih for your attention. Thank you very much. If you have any questions, feel free to address them to christian.borzak@draeger.com. Thank you very much.
Thank you very much, Christian, I think most MLTs will love this idea now that they don't need to do the normal invasive test of pricking the patient. During this pandemic, while tackling the coronavirus, we must also remember to save our babies, our newborns. And it is wonderful to see that the commercial sector has piggybacked on the drive-through concept of COVID-19 sampling. I think it's a fantastic idea and really is in line with reducing or minimizing infections among the healthcare workers, the parents, and the baby. And also it helps in the practice of social distancing. Thank you again. All right, now we are opening the Q&A to all the participants. If you have not posted your question, please do it now at the Q&A box and not in the chat. I repeat, post your question now at the Q&A box. Okay, so far we have got a few questions coming in. Just give me a minute. All right, this question is from Malaysia, and the question is, and this for you, Havinda, since saliva sampling is non-invasive and easy to self-administer, any comparison study between saliva and other nasopharyngeal swab comparison been done in Malaysia? Is there any paper?
Yes, there is two research ongoing. Another one pending for the National Medical Registry to approve, as well as the Ethics Committee. But the thing is, as we all know, there's already a USA FDA approved emergency testing for saliva. So, but in Malaysia, we are currently doing the testing, but it is still under research and it hasn't been published as of yet.
Okay, the next question is also for you from Indonesia. In your experience, among all the other alternative specimen collection, which is better for diagnosis of COVID-19?
Which is better has to be coming from WHO recommendation. As we all know, currently WHO recommends lower respiratory samples like BAL, sputums, as well as upper respiratory like nasopharyngeal, oropharyngeal. Unfortunately, these sampling methods are a bit invasive as well as aerosol generating, but they are highly recommended by WHO.
Okay, our next question is for Dr. Zainun. We run RT-PCR tests by using different kits, so different optimization done. The question is how we want to make the report and comparison data acceptable worldwide. Dr. Zainun?
Okay, so in our experience, we usually we usually acquire commercial kit. So we ensure that the kit has already been optimized for use on our PCR platform. However, we also do validation on these kits. So what we did, we usually for PCR processes, we do the analytical sensitivity where we use we use serially diluted sample with known concentration, and then we determine the limit of detection and then see whether it is agreeable to the manufacturer claim. We also do repeatability of the CT values between replicates and also reproducibility by different operators. We also do comparison using sample, known sample, for example, EQS sample. So we actually made a report for each individual kit using this same format.
Okay, here is another question for you again. Did the laboratory staff get infected while processing the samples in your place? Because I think this is a main concern among all those laboratories working here, right?
Yes, correct.
Go ahead.
To answer the question, no, we haven't. Our staff have not gotten any infection from the COVID-19 testing. I think this is due to the several We ensure our staff are well equipped with proper PPE and ensure that they are must fit tested. We also ensure their safety practices are regularly audited when they're actually working during during the PCR process.
Okay, this is a question for Christian.
Yes.
The device that you mentioned, is there any radiation effect?
Mm-hmm. I mean, of course, we are using high intensity light to measure this. So therefore, we highly recommend, of course, to keep it away from the eyes. So therefore, this is the one thing to keep in mind. Apart from that, there hasn't been any radiation effect. But of course, with very bright lights, of course, we need to stay away from the eyes.
Okay, also another question for you. Is it compulsory to determine the method comparison between the TCB measurement device and the bilirubinometer before putting the TCB into use? And if yes, how frequent it is to recommend to carry out this effort?
I think there are two paths to this. So in general, it's important on a daily basis to check whether or not the light source is still fully functional with this light check. All you need to do is it's an inbuilt optical path where you just basically take the device and push it onto the testing. It's included in the device mount. And then you know already if the sensor and the light source are calibrated. But in general, when you're introducing TCB to your institute, in the very beginning, we actually recommend to have both TCB and TSB measurements, simply to just develop a good understanding, a good feeling for the offset between those two values, to have enough experience and then deciding which are the thresholds you want to determine or define for the TCB measurement. So this is why, in general, we would recommend, at least for the first 20 to 50 measurements, to note down both values. And of course, to make sure that the same workflow is being used for those two measurements, so they are reliable and comparable. So make sure that you decide either to use the forehead or to use the sternum, and stick with it. And of course, to make also sure that the handling of the blood sample is adequate, and of course also the heel prick is done very shortly after the actual TCB measurement.
Okay, at this stage, I would like to encourage all the participants to stay until we finish this webinar, because there are some other things which we need your participation. In the meantime, I would like to direct this question to Tintun. What is your advice on handling coronavirus samples? Should it be carried out in a BSL-2 or a BSL-2 plus or enhanced BSL-2 lab? Thank you.
Yeah. For regard to handle COVID-19 sample, there's a two types. If you are going to culture or isolate or handling live culture, you must use BSL-3 facility as well as BSL-3 practices. But if you are going to conduct serologic tests or histopathology tests, or these tests, if you want to do, then you can BSL-2 facility you can use, but you need enhanced biosafety practices. For example, the access must be very strict. The access to the laboratory must be very strict. All the procedure steps must be done inside the certified BSc, and then with product, and then disinfect them as a properly used. Then this is enhanced procedure need to be applied. That's why we call it BSL-2 plus. That is enough for serology and other things. Thank you.
So thank you. So MLTs, please note that you need not use a BSL-3 unless you want to do culture of the virus, right? just for the samples, the BSL-2 plus is enough. And in fact, you should, if you are still using BSL-2, you should actually migrate to BSL-2 plus.
Yeah.
Okay, Havida, I've got a dying question for you.
Yeah.
Is there any local institution attempting to make a vaccine in Malaysia?
Yes, if you would have seen, there was 2 newspaper articles published in March as well as April in NST, where UITM as well as UM are trying to do the CRISP gene detection method in order to come up with vaccines.
That means you say that we already have the facilities to produce vaccine.
We are
research
working,
is different from production, no? Do we have?
Production, production terms, yes, because they're dealing with different businesses like pharmacogenetics.
Okay.
So there are plans, but whether it materializes or not, well, it depends.
Okay, Dr. Zaino, this is for you. Did you encounter any RT-PCR contamination during this period?
Oh, yes.
Because I think this is a very important question that actually will make us throw away all the tests.
Yes.
Right?
So we have a few occasions where we had a contamination in our PCR process. So so what we had to do is we had to stop for a while the testing and clean the lab, and then try out with negative control and see whether the contamination still exists. So I think most of our contamination came from the pipettes as well as from our manual extraction methodology.
So how long does this period take, the cleaning up?
Probably just half a day.
Half a day.
Yeah, so because we can't stop testing, so we just usually take a few hours to clean up and start back straight away. So that is from our experience.
I remember that you have experienced some reagent shortage during this period, right?
Yes,
Okay.
Especially the extraction kit.
Okay. The question here is, would you advise on sample pooling if you are faced with such a situation? I think usually when we have such situation, we always think of sample pooling.
Oh, definitely not. I would not advise any sample pooling at all, because especially during the outbreak, you know there will be a lot of contact tracing where most of it will be positive. So you have to, once you pool, and you have to go back and test back all the samples individually. So I don't think pooling is a good idea at all.
But I think in a very dire situation like in the US where you don't have enough reagent, I think this is what they do, right?
Oh.
And we actually did this also during HIV time. You know, we do a lot of sample pooling. Yep. I think that's all we have. The questions from the participants. And I would like to make a summary of the event. I think all of us know this guy, Dr. Anthony Fauci. He's the director of the National Allergy and Infectious Disease, very prominent during this time. He said, the more we learn about the novel coronavirus, the clearer it is that COVID-19 isn't going away anytime soon, if at all. So I think this is a very important statement that we must remember, that we have to deal with this for a long time to come. All right. So we may be expecting another surge again. And I hope that all the labs will actually get prepared. All right. So it is now very clear from the lectures that the clinical laboratories as well as research lab do play an essential role in this pandemic crisis, especially in areas like patient screening, as you have heard, and the diagnosis, monitoring and treatment, and epidemiological recovery and surveillance. Thank you. Rafida?
Hi again. Thank you, Mr. Woon, and all our panel of speakers for the very interesting sessions. We hope that everyone have benefited something from today's sessions and thus worth the time spent online. We do hope too that the audiences have captured the important facts presented that can help to improve or enhance their current practices or to consider some of the ideas for future planning. Perhaps for some of us, the additional knowledge obtained today would be very useful for academic purposes. Just let me remind you once again, for those who wants to have their certificate and certificate of attendance for today's webinar, you can find the link in the chat box. To help us improve in organizing our future events, we would like to ask our participants to kindly spend a few minutes right now to answer our survey question by clicking the poll box on the screen. We shall give you a few minutes to complete it, and then come and join me again right after the poll session ends. Thank you very much. We really appreciate your feedbacks. Before we close the event, I would like to introduce the individuals who have worked indefatigably to make this event successful. And here they are, our wonderful team. We would like to express our highest appreciation to Mind Ray and ChemoPharm for their invaluable support. It has been a great experience collaborating with everyone from ChemoPharm Malaysia and all our distinguished speakers. And so I'm saying it once again, and on behalf of the webinar organizing committee, thank you very much from the bottom of our hearts. The next AACLS live webinar will be hosted by our dear friends from Philippines Association of Medical Technologies, PAMET. It is scheduled on the coming 27th of June, but do look out for the official announcement when all information have been confirmed. Ladies and gentlemen, we have finally come to the end, And we would like to apologize for any shortcomings if there's any. And until we meet again, I'm Rafida Muhammad on behalf of the organizing committee saying terima kasih. Jumpa lagi. Bye.
Bye, everybody. Thank you very much.
Bye.
See you next time. Bye.